guys, this is Sai Kiran. Welcome to our lovely pharmacy channel. Today our subject is pharmaceutical biotechnology, which is here in B.pharmacy 6th semester. So first of all, we go through it. What is biotechnology and its various developments and historical developments. First, biotechnology. Biotechnology is biotechnology. Biotechnology is subdivided by three words: bio, techno, and logy. What is bio? Bio means biology or living organism. Technology, technology. Logy means study or science. So biotechnology is a branch of science that uses biological source or living organism as a raw material to produce new products. Here, what are raw materials? Raw materials include microbes, plants, and enzymes. These are the raw materials used as a living organism or biological source to produce new products. Here, what are new products? New products come under antibiotics. Antibiotics or such medicines, antibiotics or medicines. Next, development of biotechnology. It comes technology is developed with three stages. Next, development of biotechnology. It is divided by three steps or three stages. The first, before 1800s. Next, second one, classical biotechnology, and third one, modern biotechnology. First we go through it. Before 1800s. First, what is happening before 1800s? A scientist named Karoli Erecki. He introduced the Karoli Erecki scientist. Karoli Erecki is a scientist who introduced who used the first word of biotechnology. Means he used the first word of biotechnology, and he is also called father of biotechnology. In 1919, he used the word of biotechnology. In this 1800, various types of food and fermented fermented medicines or beverages are introduced. What are the various types of in food? In food we undergo. Next, in food means domestication of crops. Like crop rotation technique, mainly crop rotation technique. Domestication of crop means crop rotation technique is used, or crop rotation technique is introduced in 1800s. Why the crop rotation technique is introduced? Crop rotation technique is mainly to increase the yield value. In crop rotation, mainly yield value. To increase the yield value, crop rotation technique is used. Next, in fermented beverages. What are fermented beverages? Like alcohol beverages. Alcohol, then curd, cheese, and few other medicines, and few other foods are introduced or produced in this. In 1800s. Next, classical biotechnology. What is classical biotechnology? In classical biotechnology, In classical biotechnology, second one, classical biotechnology. This was classical biotechnology is after 1800s. Classical biotechnology introduced or after 1800 or middle of 20th century. Next, in this era discovered nucleus in cell, discovered nucleus in cell, and law of inheritance. Next, antibiotics and vaccines. నెక్స్ట్ ఇన్ 18 మెయిన్లీ డిస్కవర్ న్యూక్లియస్ ఇన్ సెల్, అంటే సెల్లో న్యూక్లియస్ ఉంటుందని డిస్కవర్ చేశారు. దాని తర్వాత లా ఆఫ్ ఇన్హెరిటెన్స్. నెక్స్ట్ ఫస్ట్ ఇన్ దిస్ ఎరా లా ఆఫ్ ఇన్హెరిటెన్స్ వాస్ డిస్కవర్డ్. లైక్ వి నో ద మౌస్ ప్రాసెస్. ఐ మీన్ అంటే ఒక ఎక్స్పెరిమెంట్ ఉంటుంది కదా, ఇట్లా మౌస్ టేల్ కట్ చేసి దాని ఒక కొన్ని జనరేషన్స్ వరకు, ఒక 3-4 జనరేషన్స్ ఆర్ 5-6 జనరేషన్స్ వరకు దాన్ని ఇట్లా కపుల్ చేసి దాని తర్వాత నెక్స్ట్ జనరేషన్ టేల్ వస్తుందా రాదా అనేది ఒక లా ఆఫ్ ఇన్హెరిటెన్స్. అది లా ఆఫ్ ఇన్హెరిటెన్స్. ఐ హోప్ యూ గైస్ నో వాట్ ఈస్ మై లా ఆఫ్ ఇన్హెరిటెన్స్. నెక్స్ట్, యాంటీబయోటిక్స్. నెక్స్ట్, డిస్కవరీ ఆఫ్ యాంటీబయోటిక్స్. Antibiotics also were produced or introduced in this era in classical biotechnology. Next, vaccines are also discovered by different scientists, like Alexander Fleming was discovered vaccine like penicillin. Alexander Fleming discovered penicillin. How he discovered penicillin? From penicillin notatum, he discovered penicillin vaccine. Like this few other, oh my god, like this few other scientists are also discovered antibiotics and vaccines in this classical biotechnology. Next, third step, modern biotechnology. In modern biotechnology, DNA What is DNA? DNA is a deoxyribonucleic acid. DNA is deoxyribonucleic acid. The DNA was discovered. The scientists told that the genetic information, the main thing is genetic information, gene code, oncogenes, and oncogenes. Next, human papillomavirus. Next, the scientists told that the genetic information, the DNA will carry the genetic information. Next, gene code. The DNA also carries the gene code and oncogenes. These oncogenes are divided, or these oncogenes derived from the normal genes. These oncogenes also causes cervical and throat cancer. How these causes cervical and throat cancers? By the mutation. By the mutation or cell division. These when these are exposed or increased in number, these causes cancer, cervical and throat cancer. Next, human papillomavirus. For this human papillomavirus, the vaccines for this human papillomavirus, the vaccine was introduced. In modern biotechnology. Restriction enzyme. The first restriction enzyme was also discovered in the modern biotechnology. Okay, I hope you guys clear up till biotechnology. Biotechnology is a study of living organisms that produce new products from the biological source or living source. Here, here what are raw materials mostly microbes, plants, enzymes. From these, the new products will be produced. Antibodies and medicines. Okay, next development of biotechnology. Before 1800s, development of biotechnology was three stages. Before 1800s, classical biotechnology, modern biotechnology. In classical biotechnology, Karoli Ereki was a scientist who discovered or introduced the word of biotechnology. Next, in this mostly fermented crop rotation techniques and fermentation. By the process of fermentation, the beverages and food materials which are comes from the fermentation are also discovered. Next, in classical biotechnology, this is after 1800s to this after 1800s century. In this era, discovered nucleus in cell, law of inheritance, antibiotics, vaccines. The first vaccine was discovered by Alexander Fleming from the penicillin autotome. Next, in modern biotechnology, this is after modern biotechnology, mainly the DNA. The scientists proved that DNA consists of double helix structure, double helix structure, DNA is a double DNA is a double helix structure. Sorry guys, a little bit. Okay, double helix structure. Next, genetic information where the DNA. The scientists told that DNA के the genetic information, gene code, uh, genetic information and gene code. Next, oncogenes are divided from the normal genes. Next, discovery of vaccine for human papilloma virus. Okay, I hope you guys clear until here. We go through it. Okay, various domains in biotechnology. What are the various domains? White biotechnology, white biotechnology, white green, red, dark, uh, brown, violet, gray, gold, and blue. 1 2 3 4 5 6 7 8 9 10. Okay. Next, various domains in biotechnology. In this era, or various domains in biotechnology. BT stands for biotechnology. Don't think it's a different. Okay. First, what is white biotechnology? White biotechnology is mainly for industrial 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 development of biotechnology. White biotechnology is used for industrial development. Next, green for agriculture biotechnology. Green for agriculture biotechnology. Medicine and human health. Medicine and human health. Next, dark biotechnology. Bioterrorism and weapons. Next, yellow, food and nutritional, for development of food and nutritional biotechnology. Brown, deserts. Violet, mm, the violet mainly stands for ethical and philosophical biotechnology. Gray, protection of environmental. Next, gold, Gold biotechnology used for bioinformatics. Bioinformatics. Next, CS and computer science and chip technology. Chip technology. Next, blue for marine. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, these are the various 10 different domains. Various 10 different domains are introduced in biotechnology. White biotechnology for industrial development. Next, green biotechnology for agriculture biotechnology. Next, red for medicine and human health. Next, dark is for bioterrorism and weapons development. Now next, yellow is the yellow biotechnology stands for main food and nutrition. Next, brown, the desserts are mostly brown, right? That's why uh, brown biotechnology is for desserts. Next, violet, ethical and philosophical biotechnology. Gray is for protection of environmental. Next, gold, bioinformatics, computer science and chip technology. In Robo movie, we can see the chip technology, right? The Professor Boro introduces red chip into Robo or Chitty. Okay, that is chip technology. Next, blue for marine source. Marine source. These are the various domains. Next, objectives. Objectives of biotechnology. What are the objectives of biotechnology? The objectives are nothing but to produce safety, efficacy, economical. Safety and efficacy and economical means cheap price. The poor people or middle class people are also able to purchase drugs. That is called economical. So safety, efficacy and economical to produce safety, efficacy and economical drugs, and also to produce new discovery means the drug should not be discovered before, or means it should be clear. Means a particular person means the he should own the own patent, so he should not copy from any others. The main thing is the second, new discovery or new design. While discovering any drug, it should be not prepared, or it should not introduced before. It should not before. I need to the design should be easy, I mean to look or to attract the people who cannot buy particular tablets or capsules. When the size is elegant, I mean not elegant, when the size is not elegant, people or patients are unable to take those. So the design also must be categorized here. So that is new design. Next, applications in this antibiotics. The applications are antibiotics and vaccines. Discovery of introduction of antibiotics and vaccines. Antibiotics, vaccine. Vitamin B12, antibiotics, vaccine and vitamin B12. The vitamin B12 tablets or capsules are introduced. Next, human gene therapy. Next, automated bioscreening. Automated bioscreening methods. So okay guys, these are the objectives and applications and various domains which are introduced or which are developed in biotechnology, right? Okay guys, thank you.